Well, amino acids are simple components of almost everything, let's say. So in nature, proteins and peptides, which are molecularly, which has biological activity and very many implications, are molecules of life. So they are composed of amino acids, and all natural amino acids have the L configuration, and 22 of these natural amino acids, because there are more natural, but usually we consider this as proteinogenic amino acids. So the 22 amino acids which enter into proteins after the system which life uses for building proteins. Then there are others which are modified after this, and they have special functions. So amino acids are the constituent of nature. They are usually found in nature. They can be produced. All the 22 amino acids are produced in the L form, which is the mirror image of the D form. So this will require maybe to specify what is chirality and what are chiral molecules, So I don't know if you want me to explain this, because in nature these molecules are chiral. So it means that they are made like this end, and you will not find this end. So if you have a glove and you want to use the right glove with this end, it will not fit. Okay, so this is the principle of chirality. So all these amino acids are, let's say, in this way, and they don't exist in this way. Okay, so this is one fundamental principle of how molecules are replicated then in nature. So the reason why we want to, for instance, to study non-natural amino acid, or non-natural maybe because they are of this form, or maybe because they are made like this instead of like this. Okay, so this is due to the fact that many drugs contains amino acid. Okay, so drug discovery goes through the use of building stones, which might often be amino acid. And many of these drugs, for instance, are devised as enzyme inhibitors. So the most common enzyme in nature is protease, which split proteins. So if you use a peptide that you want to use as a drug, it might have a very good activity. But if it's done by natural amino acid, it will be split immediately in nature by proteases. So if I put inside an amino acid which is non-natural and will not be recognized by this enzyme, then I will prevent that this molecule will be destroyed. Okay? So this is one general principle which brings a reason why there is so much interest in non-natural amino acids. So I would say that this is a very important principle. So there is a lot of interest, and company who are after drug discovery, they try all kind of modification of this amino acid in order to have molecules which are resistant to proteases, and they will not be destroyed. Then they have to have the activity that we would like to have, and then this is another point. So this goes with drug discovery, but in general, amino acids are done for this. So a natural amino acid, they are put into molecule in order to prevent their hydrolysis by proteases. Okay, so there are many methods where you can make these molecules, and this conference is showing that the use of enzymes as catalysts is important for making this kind of molecules. Also because many of these are chiral anyway, so that we need to have a mean to introduce this difference between the two ends, which cannot be done by chemical reaction, conventional chemical reaction. It needs to be introduced by proteins, which are intrinsically dissymmetric.